Hello friend, welcome you all to my channel Learn Innovative with me. My name is Sasi Usar Chauhan. Today I am going to show you that how we can analyze your real-time gene expression data and represent in the form of cycle over endogenous control or in the form of delta CT value. But if you want to learn how to analyze and represent the data in the form of whole chain expression from the real-time gene expression data, you can see my previous video tutorial. But today I am going to show you that how you can analyze and present your data in the form of cycle over endogenous control or in the form of delta CT value. Here is the data of an gene expression of real-time PCR, in which you can see there are two group control and treatment having 10 sample for each group, and the endogenous control gene is 18 years, and the sample was run in duplicate. You can see the CT value in duplicate for control as well as treatment group, and the target gene or gene of interest is lag3, and the CT value of lag3 duplicate here. So for the analysis of this data, first we have to calculate the CT mean of endogenous control as well as target gene. For the endogenous control, select the well equal to CT value 1 plus CT value 2, just enter, select the well. If you see the plus sign, just drag it to all the sample. You will get the CT mean for all the sample for 18 years. Like this, for the leg 3 equal to T value 1 plus 2, enter, drag to all the sample. After calculating the CT mean of endogenous and the target gene, we have to calculate the delta CT value. First, I want to show you that what is the delta CT. You can see on my laptop screen an amplification plot of an gene experiment. Here is the 18 years CT value, that is 11 for the housekeeping or endogenous control gene, and the target gene for the same sample, the CT value is 29 for the leg 3. When we subtract the endogenous gene CT value from the target CT value, that is known as delta CT value. So you can see here cycle over 18 years or endogenous control, or delta CT is equal to target CT value minus endogenous control CT value. Here we have to calculate the delta CT value. For this, equal to CT mean of target gene minus CT mean of endogenous control gene. Enter, select the well, drag to all the sample. We will get the delta CT value for control group and treatment group. And to represent these data in the form of graph, here I am opening the GraphPad print software, select the column, rename the group. In my case, group 1 is control and the second is treatment 1, and copy and paste the data of delta CT of control and treatment here. For this, delta CT of control and the delta CT of the treatment group, paste in GraphPad print software, click on data in the graph, and here you can select the graph type according to your choice. I am selecting individual value, showing the in a scatter plot, showing the median. You can select. Here is the option to select various things like BSD or what you want. You can select from here and just playing with the median. Click on OK. Here you can rename the Y title cycle over 18 years or lateral CT. As we know that in real-time gene expression data, CT value is inversely proportional to gene expression, means if the CT value is high, the gene expression is low, and if the CT value is low, means gene expression is high. So we have to inverse this y-axis so that we can represent our data. To reverse this axis, just double click on the y-axis, and you can see here it is selected on left y-axis. In standard, just click here and you can see here is the option to reverse the axis. Just click on the reverse and apply and click on OK. You can see the y-axis is reversed and the data showing the gene expression of leg 3 is high in case of treatment 1 group. So you can see data analysis of real-time gene expression in the form of cycle over 18 years or in the form of delta CT value is very easy. I hope you like the video. If you like the video, please subscribe my channel. Thank you.